I work mainly in vitro with membranes. I'm doing mainly membrane biophysics, but here in Germany, the project is try to understand what happens when biomolecular condensates interact with membranes. So I will show a short introduction on what are biomolecular condensates and why it's important to study the interaction with membranes. I will provide some background on what has been done here in the lab of Rumiana Di Moda, where I'm working. And then I will briefly show some results on how these condensates can wet and remodel membranes at the microscale. And then I will focus on how to use these environmental sensitive probes that you already know to study membrane condensate interaction. So this is the image of the cell that we had 18 years ago, 15 years ago. So we have a cytoplasm enclosed in a membrane, and inside it we have several membranes surrounding organelles, membrane-bound organelles. And until this work appeared in Science in 2009, and I don't know if I can play the video here. Let me see. I never know how to play the video. Okay, but this work showed these tiny organelles that are liquid-like, and they don't have a membrane surrounding them, and they behave like liquids. They can fuse together, they can appear and disappear, and this revolutionized the field of biology, because nowadays we have a more complex image of the cell. It's full of droplets on the inside, and each one of these is an organelle. Some of them are transient, like stress granules. Some of them are there always, like the nucleolus. And the idea is to try to understand the interaction of these things with membranes, because it's logical that in such a crowded environment as the cell cytoplasm, this droplet can encounter the membrane. So the membraneless organelles can interact with the membrane-bound organelles that we know before. And there are several examples of this already. So this is, for example, these are vacuoles of Arabidopsis, and inside they have this storage protein that phase separates and can wet the membranes of the vacuoles, and it can remodel these membranes, and this is an important step on the plant embryogenesis. Another example that is very different is this on the synapses. So it is believed that this droplet is a synapsin mainly phase separated that has inside these synaptic vesicles, and a lot of examples keep appearing on different functions. For example, this interaction between droplets and membranes are critical for tight junctions and regulates autophagy and so on. So this is what we will try to understand. And let me tell you some background on what has been done in the lab here before. So here they worked with these vesicles, giant monomer vesicles, that inside have a mixture of polymers, PEG and dextran. And this polymer mixture can phase separate when you increase the concentration of these polymers. And the way of doing that inside the vesicle is by increase the external osmolarity. So the water comes out of the vesicle and the polymer concentration increases, and you can generate phase separation inside the vesicles. And as you can see, if you keep doing this, you can generate some morphological transformations. And they have been studying this for a lot of time, even before the phase separations were discovered in cells. So it's really nice to see how working in vitro can give some interesting results and can sometimes predict things. And here, Di Moda and Lebowski described for these systems a wetting transition. And this means that in analogy to what happened when you have a liquid droplet and a solid support, that depending on the surface tension and the interaction between the droplet and the support, the droplet can spread more or less. The same happens when these droplets contact now the soft substrate that is the membrane. And if you can measure this contact angle, you can have an idea of the interaction. So this has been done in the lab before, but in my project, what we wanted to learn is what happened with protein. Now, protein condensates interact with membranes. And for that purpose, we used glycinin, that is one of the most abundant proteins in the soybean. It's a storage protein. And the nice thing about this is that glycinin phase separates in the presence of sodium chloride. So if you see here in this diagram, at low sodium chloride concentrations, we have an homogeneous phase, then we enter a phase separation region, and then at higher concentrations, again, we have an homogeneous solution. So it's a really simple system to use as a model. And on top of that, the mechanism of phase separation has been explored before in the lab. So we use these droplets, put it together with GUVs, and this is what we got. So at very low salt concentration, there is complete dewetting, meaning that the droplets do not interact with the membrane. If you increase the salt concentration, you get more spreading of these droplets on the membrane, meaning that the interaction is increasing. And you can see also that in some cases, the membrane points towards the condensate in the region of interaction or towards the interior of the vesicle. So you have different affinities. And if you keep increasing the sodium chloride concentration, you can have the complete wetting or spreading of this droplet on the membrane. And what you can do is you can measure the contact angles using microscopy. And these contact angles here are related with the different interfacial tensions in the system. And Lipovsky derived this factor that is called the geometric factor that allows us to quantify now these different morphologies and the different interactions between the droplets and the membranes. And this is the last thing I want to show you. Something really interesting that we saw is that when the vesicles have enough excess area, so when there is enough membrane excess in the interface between the condensate and the membrane, we observe this interfacial ruffling or fingering that depends on the excess area, because when we deflate the vesicles, meaning that we create excess area, we see more of this phenomenon. And when we inflate the vesicles, meaning that we make them tense, we see less of these things happening. And this is more obvious if you use a micropipette and you aspirate the excess area, you see the smooth of this interface. And if you release the tension, you see again the curves on the interface. And these are just more nice pictures. And here, what you can see is that it's a mutual remodeling process. The membrane and the condensate get reshaped and in different ways. This is a 3D reconstruction. And by using 3D STED, you can remove out of focus light and really see that these structures are tubes, for example, and you can quantify their dimensions. So these are previous results, and this is a manuscript that we have already submitted. So we learned that the condensate can wet the membranes, and you can tune this wetting by changing the sodium chloride concentration or also by changing the membrane composition. I didn't show that. And something interesting is that the membrane dynamics in this region of the membrane that is interacting with the condensate are reduced compared to the rest of the membrane. And in order to learn what is happening there, is that we go to the second part, that we will use these environmental nanosensors. I don't need to tell this again because it has been told already by several speakers, but these are the group of probes designed by Gregory Weber, the drum probes. Agdan is the soluble version, and Lordan is partitioned to membranes because it has a Lauroid tail here. And these probes respond to dipolar relaxation of the solvent. So we use hyperspectral imaging and phasor analysis, also FLIM, And this is an example. So when you have this protein in water with Agdan at different concentrations, the solution is homogeneous, just in water. And if you increase the concentration of the protein, this is how the phasor plot, the spectral phasor plot looks like. And you can see that the pixel cloud moves towards lower dipolar relaxation levels because you are increasing the crowding. And you can do a two-cursor analysis here and have the distribution, the profile distribution of these pixel clouds. And if you take the center of mass of these distributions, you can plot it like this. And here you can see clearly when we increase the concentration of the protein, we are decreasing the dipolar relaxation. So the idea is to first to put Agdan in this protein system and study the phase separation of this protein in the different regions. So we took these compositions that you see here. So a couple of compositions in the region 1 that is homogeneous, then some compositions of sodium chloride that enter in the phase separation region, and another point here in the homogeneous phase again. And this is the spectral phasor, how it looked like. There are two main pixel clouds corresponding to the homogeneous and to the phase separated phases. And if we use a continuous color scheme to paint the images, you can already see here that even between the homogeneous phases or the phase separated phase, you can see differences because this point is not the same than this and not the same than this, and the same happened in here. So when we do the cursor analysis of this, we have the distributions, and here you can see more clearly that for the homogeneous phases, there are differences. Agdan is sensing differences between these different points, and also the same happening within the phase separation region. So we wanted to understand what is happening here. I mean, the system just consists in the protein and sodium chloride. So what has one possibility is that the protein is changing its secondary structure, and we check that using infrared spectroscopy. So this graph, you have the percentage of secondary structure for the different conditions, but just focus on this one. Here, I'm plotting the percentage of secondary structure change compared to the initial structure of the protein in water. So we're seeing how the structure changed compared to the original, let's say, the structure of the protein in water. And I'm not plotting the beta G because it doesn't change very much. But if you can see here, the blue is random coil, and it changed a lot when with the onset of phase separation, and the alpha helix content decreases, and then these values more or less come to similar values that at the beginning. So Agdan is being responsive to these changes that are happening in the protein during the phase separation process. We also did Raman spectroscopy, because using Raman spectroscopy, we can measure the water band, and the water band can be decomposed according to the different hydrogen bonds that the water molecules are involved in. And the two main bands correspond to decoordinated water and tetracoordinated water. So we did this, we measured this spectra for the different conditions, and you can see here that the water band changes in the different conditions, in presence of the protein, of course. And a way of quantifying these differences is by doing like a GP function now of these two main bands, and you can see that there is a change. So the percentage of water involved in tetracoordinated hydrogen bonds increases and then decreases. And this trend of having an increase first and then a decrease is similar to what happens with the protein structure, and also similar to what we see with the Agdan response. So Agdan is really a very nice probe that can sense these changes happening in the phase separation process. And to finish, let me show you what happened to the membrane. So you know that Lordan can detect changes in the phase state of the membrane not only between liquid and gel phases, but also intermediate hydration states. So it's very sensitive. And what we did was we labeled the membrane with lower than. The condensate is not labeled. And you can see that with phasors, we can separate now the contributions of these two parts of the membrane, the one that is in contact with the condensate from the rest. And what we saw is that the part of the membrane in contact with the condensate is more packed than the rest of the membrane. Next, we wanted to know what happened now when we increase the wetting, meaning that we are increasing the interaction of the droplet with the membrane. And this is the result. So when we increase the wetting, so in this case, by increasing the sodium chloride concentration, we increase more and more the membrane packing. So that part of the membrane gets more packed and dehydrated than the rest of the membrane. We were wondering if this is just something that happens in this specific system or was something more general. So the next thing we did was to evaluate another system that is the one that I showed you at the beginning, the vesicles containing the protein, the polymer that phase separate when increasing the osmolarity outside. So here, this is what happens. So in the first step, you don't have phase separation. You only have some tubes formation in the interior of the vesicles, but then you have phase separation. And we don't care about the morphological changes. We are only interested in seeing what these wetting changes are doing to the membrane. And for this system, we use film because we needed to collect more photons, and film allows you to accumulate frames. And these are the results. So what we see is again the same. When the system is phase separated and we are increasing the wetting, we increase the membrane packing in that region of the membrane. And the same thing that happened with glycine and the CUVs, we measure it again with film, and we obtain the same results with a spectral imaging. And with film, with film, you can separate the contributions from the polarity and the dipolar relaxation. And here is the dipolar relaxation for the two systems. And in both cases, the dipolar relaxation follow the same frame than the polarity. So the membrane is becoming more packed and more dehydrated when you increase the wetting of the system. So we showed that Agdan, together with hyperspectral imaging and the phasor analysis, is a very powerful tool to understand phase separation, and it gives you a fingerprint of a condensate in a given state. And with Lordan, we learned that the part of the membrane that is in contact with the condensate is increasing the packing, is more dehydrated than the rest of the membrane. And